You're listening to KUCI 88.9 FM in Irvine.
The views and opinions expressed on this program do not necessarily represent those of KUCI, its management, or the UC Board of Regents. For more information on this or other KUCI programs, visit kuci.org or kucitalk.org.
Welcome to KUCI 88.9 FM. If you're here at 4:30, you're here for office hours with Newton Hood. Today we have a special guest. We have Lydia Dean. She's a PhD candidate here in the Department of Ecology and Evolutionary Biology at UC Irvine. Lydia, thank you so much for stopping by office hours.
Thank you so much for having me.
No, I'm so happy to have you on. You do such cool work, and I can't wait to dive into the research you're doing here at Irvine.
Thanks. Yeah, I'm looking forward to it.
So first, I'd like to get everyone's origin story about how they became a scientist. So we all start somewhere. So for yourself, did you always know you wanted to become a biologist? Was there a particular moment that got you on this current journey?
Yeah, I didn't always know. So I started off kind of like end of high school, undergrad, knowing that I liked biology and was interested in that realm. And I started off wanting to be a vet. So I loved animals and I was doing all this stuff with animals. And in my sophomore year of college, I went on a study abroad trip to Costa Rica.
Oh, nice.
And we took a course beforehand in tropical biology. And then we went and spent 3 weeks there. And I learned ecology and I learned research. And after that, I was like, oh my gosh, this is what I want to do. And particularly in Costa Rica, I learned bird watching. I just that was something that was taught to me. It was never something that I knew was a thing or explored before. So in the 3 weeks, I ID'd 150 bird species. It was
impressive.
A real deep dive. And then ever since, I've been an avid birder. I got into research, and that kind of catalyzed everything, I would say.
So what got you to do that study abroad? Did you always wanted to do it, or was there something that got you to sign up for it?
I think it was an advisor of mine who recommended I take it. And it was kind of one of those opportunities. I went to a really small school, so it's something that everybody knew was a thing, but not everybody took it, right? It was the people that liked biology and ecology and that kind of stuff. But my advisor was like, this would be a great opportunity for you. And I was like, well, yeah, sure. I want to go to Costa Rica. And then, yeah, that's how I got there.
Oh, shout out to the advisor.
Yeah, absolute shout out to my advisors in undergrad.
Yeah, people took those meetings seriously. You never know what type of nuggets of information you'll get.
Yeah, absolutely.
So you did this awesome study abroad program that shifted you from veterinary medicine to biology and ecology. So how did you make the decision to go to graduate school? Did you always know graduate school was an avenue that you could explore, or was there something that got you interested in graduate research?
In that study abroad trip, I called it an epiphany moment. It was when I knew that I didn't want to do vet school and I wanted to do research. So I would say knowing that the research was the avenue, that was maybe the moment that led me towards grad school. But once I knew I wanted to pivot towards research, I got involved in a research lab and then learned what graduate school was. And then from there was slowly advised into pursuing graduate school from there.
What was your undergraduate research experience like?
It was really nice. I worked for 2.5 years in the same lab. And it was like a joint lab. So I
Mm-hmm.
got like 2 different lab experiences, 2 different PI experiences. And yeah, the lab was great. The research I was doing, I loved. And then it was like a really supportive environment. So they really encouraged me to pursue grad school.
Oh, that's amazing.
Yeah.
So how was your search like? Because I know for everyone, their grad search takes a different form.
Mm-hmm.
So how did you end up at UC Irvine?
Right.
And how did you end up picking the advisor you ended up deciding on?
Those are good questions. So I did the standard route. So in the summer before the fall of applying, I was searching through different websites and looking at different PIs who I found like their research interesting. So I had like a big spreadsheet. I was one of those organized people who had a big spreadsheet of like PI, whether they were taking students, kind of their research topics, whether they had funding, and then narrowed it down to a list of like 15 or 20 that I wanted to email. And then I met with, I think 3 or 4 of them, like in a Zoom interview. And then I applied to 2 PhD programs and 1 master's. And the master's didn't have funding, and then I got into both of the PhDs, and then decided on UCI.
Hey, congratulations!
Yeah. So that was my process for like looking for grad schools and that kind of stuff. I knew I wanted to. I did my undergrad in Wisconsin, so I was kind of a bit picky on going to a place that didn't have snow. So that had a big influence. So I was narrowing down by place and then by people, and then eventually got here.
Oh, that's awesome. Yeah. No, weather is an important metric.
Yeah.
Because you're going to be living in that place for several years.
Yeah.
You have to be able to enjoy actually living in that area.
Right.
So I fully agree. As someone from the Midwest, I also
Yep.
do not ever want to see snow near me ever again.
I like snow, but I don't like driving in it, and when it gets all the roads dirty.
I like
and,
seeing snow. Like seeing the snow caps is
Yeah,
awesome.
yeah.
Like walking through
Yeah.
it, and I'm done with that. I can't do it anymore.
Yep.
So how did you specifically find your advisor? Was it from reading? Did you just find their website on their faculty page, or was it through word of mouth, or how did they get on your radar?
Yeah, great question. So they did research that was a little different from what I did in undergrad. Half of the people I was looking at were, I guess you would say, like collaborators on papers who I kind of knew did similar research. And then other people were people that did not have any kind of like mutual connections and were like very much random people that I just found online. So Kalen was one of the ones that I just found his website, or I think I was looking through papers on Google Scholar, papers that interested me, and then found the authors, and then like did like a little backtrack of finding where that research was going on. So yeah, it was through papers, and then I found his website, and then just like cold reached out, and it kind of worked out from there.
Oh, that's awesome. I think you're the first person that I've asked that did like the researching papers in the field that you want, and then contacting those professors. That's awesome.
Yeah, I will say a lot of people I think that I hear tend to have that like mutual connection to some extent, and I think he was like maybe on a paper that I don't even think there was any connection to the prior work that I did. It was more just like the way that the theory kind of led me to him.
Oh, I'm glad that worked out. That's awesome.
It was cool.
So can you tell us, talk about the focus of your dissertation, the type of awesome research you've been doing while you've been here?
Yeah. So I super broadly am interested in like plant herbivore interactions. So the way that arthropod herbivores interact with plants, how that shapes the community dynamics of arthropods, how that shapes plant defenses. And then more specifically in my research dissertation looks at indirect defense of plants, which is like a form of defense that plants have that recruit, that's not directly defense from the plant itself, and how birds play a role in this. So the plant traits that mediate indirect defense, allowing birds to play a role in defense against herbivores.
So when you say plant defense, can you talk a little bit more like what type of defense mechanisms plant have? So I feel like for myself, and I think most people think of like thorns.
Yeah.
Like that's the first thing we think of when we see plant defenses. Yeah. So what other types of ways can plants protect themselves from being eaten?
So I kind of was saying indirect defense. So there's direct defense and then indirect defense. Direct defense, like thorns are a great example of that. So that's one of the physical plant traits that a plant can have that defends itself against herbivores.
Mm-hmm.
Things like leaf toughness, so the toughness of the leaf, that can deter herbivores because it's not as easy to eat. And then there's also the palatability of so like the leaf chemistry that makes it more or less palatable to an herbivore. And so the chemistry typically tends to be toxic. Non-toxic versions could just deter the herbivores themselves. And so that's kind of there's physical, and then there's like chemical direct defenses against herbivores.
So for like the chemical defenses, is it when the plants make themselves either taste bitter or release some type of toxic chemical? Does it affect all organisms the same, or do they like target specific animals where it's like only caterpillars find this disgusting, but if you're a rabbit, you're perfectly fine?
Yeah, definitely. There'll be definitely some variation between, especially things like mammals versus arthropods. There'll be generalized insects and like specialized insects. So a lot of the specialized insects or like specialists can probably metabolize the chemicals and then still eat those plants. Whereas it's probably more of a deterrent for generalists that like if like a generalist caterpillar falls on the plant or gets laid there, it's probably not going to grow up very well. Whereas something you can think about like monarchs is like a really good example of this, where they do really well on milkweeds because over time they've become specialized to eat those milky leaves and with the milky sap on them. That's generally deterrent to most herbivores.
Oh, I didn't know that. Oh, that's impressive. Also, because aren't monarchs like they're bitter to like predators?
Yep. So they sequester the toxins of the milkweed that then helps them protect themselves from predators too.
Oh, nature is scary.
Yeah, very scary.
So you mentioned that these plants could also recruit birds to help with the defense. So can you talk a little bit more about how does that work? Like how are the birds being told or signaled to come to this plant?
Yeah, so that's kind of what my thesis is asking. To kind of step back a second, indirect defense is like the other half of that. And so we know that plants can recruit arthropod predators, so things like parasitic wasps. They can use cues from the plant to come find the herbivores on the plant. And the plant, and this cue is a chemical that's released from the plant, and so it comes off in like a suite of different compounds that are released. And the parasitoids are very, very hyper specialized to this and can tell even the amount of herbivores on the plant just from this like cue and this like emission of volatiles from the plant, which is super, super cool.
Wow.
And so we know this about arthropod predators. We know that they can use the plants, and what we kind of know less about is if birds can smell this.
I see. And
so there's been some work showing, like in like aviary tests where they give a control plant and then a herbivore induced plant to birds and see which one they go to first. So there's been a couple experiments showing that they do tend to go to the one that is herbivore induced, which means that the cues, or those chemical cues, have been released. But it's not really been widespread, and there's a lot of variation in what we're seeing. So that's what the main part of my dissertation is looking at, is if I can induce these chemicals in the plant and then see if birds can come to those specific plants.
So is it just the damage of the tissue from the plants that's causing the release of the chemicals?
Yeah, so typically when it's herbivore induced, it's because the herbivore is like chewing on it. There are multiple chemical pathways, so there are different types of herbivores, ones that have like sap feeding, so they have like a little proboscis. There are some that chew, which is like a caterpillar. They trigger different pathways, which trigger different volatiles to release from the plant. And there was something else I was going to say about that. What was I going to say? Even the saliva of an herbivore can trigger it, so not even like chewing and damage, which is really interesting. And then something like the poop of a caterpillar can induce it, and then even laying an egg. So if a butterfly comes and lays an egg, it triggers these chemicals in the plant.
Talk about coevolution.
Yeah, I know.
So you also said the predator could detect how many herbivores are on the plant?
The arthropod predators can, yep.
Wow.
Yeah.
So is that well understood how So I'm assuming the plant releasing a different suite of chemicals, or is it or is it like intensity, like it's proportional to the number of herbivores on the plant?
Yeah, I don't know super much about this, but I do suspect it's like emissions. So like when we look at volatile analyses, or like when we collect volatiles from the plant, things that matter are the amount of chemicals coming off, like the emission, and then the composition of those chemicals, like what is actually being induced. So I'm sure both of those things kind of affect like how much or the amount of herbivores on the plant as well.
So how do you go about collecting data for this awesome experiment?
Yeah, so there are kind of two parts to this experiment that I think of. Firstly, when I am inducing these volatiles, I don't want there to be herbivores on the plant, because the bird could smell the herbivores or see them and then therefore want to go to that plant. So I'm removing herbivores from the equation and spraying the plant with a chemical, methyl jasmonate, which bypasses the herbivore and just triggers that synthetic pathway to produce the volatiles. So that's what like the first part of my like experimental design and methods has been. And then the second half is actually seeing whether the birds are going more to those plants than the control plants. And to do that, I make caterpillars out of clay, almost like Play-Doh that doesn't harden. So I put these on the plant, and then I have mealworms that I put on the plant as well as my like assay to see whether birds are going to those plants more than the other ones. And the clay is really cool because you'll see the imprint of the bird attack on it. And then the mealworm is typically just gone, because, and like we'll put like glue at the base of the branch so that ants don't go and take the mealworm instead. But yeah, that's kind of the design setup for an experiment like this. We use chemical analyses like gas chromatography, mass spectrometry to analyze the volatile samples that I collect. So I'll place like a charcoal trap on the plant, pump the air through it.
A charcoal trap?
Yeah, it's like, I don't know how else to explain it other than this, but it's like a sorbent tube that, and charcoal is like a really good, it picks up chemicals quite well, like the volatile chemicals. So if I put a bag over the branch, I stick the tube in there, and then I use a pump to pump air through it from the plant, and then I can collect like the sample for the volatiles.
So I was going to ask you take the plant with you, so there's a way to take the sample with leaving the plant intact.
Yeah, that's really the nice part about this, is I can do all of this in the field, which is cool.
So are there particular species of plants you're working with, or are there multiple species?
Yeah, so I'm working with 3 species right now. 2 of them are characteristic of the coastal sage scrub ecosystem. And then one of them is more of like a marsh wetland species, so kind of getting a little bit of variation in what I'm looking at, seeing how widespread this phenomena is. And so 3 species of plants, and then also like generally looking at the bird community that lives in both of those habitats.
So it's not so it's birds in general as predators for arthropods.
Yeah, this is kind of the like the companion, I guess I would say, to the aviary experiments, which tend to look at like one bird species, they get to learn a lot about that species, but they don't know how generalized this is. Whereas like if I bring this into the field like I'm doing, I get to see if a lot of bird species are doing this. So I'll conduct like bird observations to see, put out trail cams, that kind of thing, see who's coming around and that. Yeah.
So how well does the clay model record data on strike attempts? Is it real easy to determine like whether or not it was a bird versus another animal that struck it?
That's a great question. Sometimes it's typically pretty easy with a bird.
Okay.
They tend to like leave a really nice V shape, and they tend to grab on either side of it, so you can tell pretty much straight away. Anything else, sometimes you can question it. If like a branch hits it and leaves like a little line, you're like, oh, I don't know. You can also tell arthropods attack on it, because you'll see where the little arthropod mouth or like proboscis has gone into there. That's not what I've been doing for this project, but for other projects I've been interested in that. And so that's like another cool way of seeing that. But yeah, sometimes it is a little tricky. And then sometimes it's really funny. You'll see one that it's absolutely destroyed. Like the bird does not get the hint that it's clay. Like there'll be like 10 or 15, like it's chewed to pieces. And it's really funny when you find one of those.
Oh, that's awesome. So the question that I have, like schematic wise, is, so like how do you position the caterpillar to like I guess you're seeing if the bird could detect the volatility of the chemicals.
Right.
Yeah, I guess, yeah. Because I was like, oh, what if the bird just never sees the, like, you have to worry about where you place the caterpillar on the plant to make it accessible to the bird?
Yeah, I'm definitely putting it on the outskirts of the
Okay.
plant. I try to put it at the same position as the controls, right? Because then they would equally be able to see and smell
I see.
the caterpillars and the mealworms. But I want to know if they're going to the induced plants more to know if it's that chemical that's attracting them there, like more, or the volatiles of the plant that's attracting them there more often.
Do you have like a team that goes out with you? Because it seems like a lot of work.
Yeah, I have a team of 10 undergraduates. I also have like another experiment going on as well for the second part of my thesis. So they're kind of using, or the team of undergrads are kind of helping out in a lot of different ways. But yeah, they come out into the eco preserve with me all the time. And I have 216 plants out there that I'm sampling. So yeah, that's a lot.
So all right, so how long does it take you? Do you have to separate all of them each session, or do some of them get left for later on?
Yeah, so I do a third of them every day.
Okay.
And I did it for a whole month.
So what?
72.
Oof.
Yeah.
It's a lot of plants.
Yeah, it's a lot of plants. So I have to go out. This experiment is quite like ritual. There's a nice ritual to it. So for about a whole month, I would go out. I would spray the chemical and the control solution on every plant. I would check if the caterpillar had been attacked, and then I would check the mealworm and then replace the mealworm. And did that for 30 days.
Wow. They can never tell you're not dedicated.
Yeah, that's the thing. I'm trying.
Oh, that's amazing. So we're coming to a close on this awesome discussion. Always ask people about what are their career aspirations and career goals. What got you here? Like what career got you here?
Yeah.
So can you talk a little bit about like where do you see, like what career paths are you looking at?
Yeah. I went into it thinking, well, I love teaching, and I have 10 undergrads for a reason, because I love teaching them all, and I love mentoring, especially getting to work one-on-one with them. So that's, I think, will at least be a part of my future, and however that looks. I went to a really small school. I went to like a private liberal arts for college, and then have now experienced a very large UC public university. And I think I've learned that when I do teach, I want to teach at a small university that I can have a small classroom and get to know all my students and work really closely with them. So if teaching's in my future, that's kind of how I foresee that. And I would like research to be a picture of that, or a component of that, but have it be like undergrad led research. So still having like a small university is great for that. On the other hand, I also love field stations and I love being at field stations, and I love the organization and the running field stations. So I think it's going to be based on like what opportunities I find at the time that I'm looking for them. I could see either of those being like something I would love doing.
No, yeah, like I fully agree. Like the dynamic, like the R2s are more intimate. Like you feel like you're making a more of a difference. Like you're actually interacting with students, like in a smaller thing, versus here's like these massive, disgusting 300-person classes.
Right.
And you just don't know anyone.
Right. And I'm like such a personable pet. Like I need to know my students to be able to teach them.
No, exactly. Yeah. And it's like, even just like I did my master's at Cal State Fullerton. So it's like, even just like the mentorship, the undergrad research culture is a lot better than it is here. There's a lot, students get hands-on experience with doing actual science, versus here it's like, good luck. If you seek it out on your own, you could find it, but we're not gonna like force you. So I understand the dilemma of like the resources at R1s are amazing, but like you feel so divorced from people here compared to other institutions.
Right. Yeah, and I think like I don't care as much to be research productive as long as I feel like I'm making a difference with like undergrads. And I think the beauty about ecology too is you don't always need a ton of resources. You can just go out and like see what's happening out there almost, and you kind of makeshift methods and supplies, I think, in the end. So yeah.
I do have one last question. Have you seen the movie The Happening?
No, I haven't.
So there's another Jasmine was a guest earlier this quarter, and I was talking about the movie The Happening, where there was these plants where people were just mysteriously dying, They were dying in the most shocking, crazy ways. But what turns out is that the plants were releasing chemicals into the atmosphere that's causing people to kill themselves. Like if people would aggregate, because it's like a plant. It was the premise of the movie is the plant make the chemical defenses to ward off against predation. So they actually made the chemicals for humans, and it was such an interesting movie that like. And I learned like plants actually do release chemicals and stuff like that. Like, wait,
biological warfare.
Yeah. So we're gonna have to have you watch that movie and get your commentary. I don't think Jasmine's seen the movie either, so we should have both of you watch
a little
it.
watch party.
Exactly, and you both could compare notes about what did the writers got right and wrong about plant chemocals.
Yeah. Because she does like plant volatile stuff.
Exactly. Yeah. So we should have both of you get some commentary on it. Yeah. Maybe invite the director or something.
That would be awesome. It makes me think of The Last of Us anytime I think of like.
It's the same thing.
Yep.
Lydia, thank you so much for stopping by Office Hours. We have to have you on again.
Thank you so much for having me. This was really fun.
No, anytime. And thank you all for tuning in to Office Hours, and I'll talk to y'all next week.
What do your children know about alcohol?
Makes people do bad things.
Act stupid and do silly things.
Alcohol makes you fight people.
People won't respect you. Personality change.
Can't think straight. Stinky breath.
Listen to your children. Talk to them in a way that they understand. This message was brought to you by the U.S. Department of Health and Human Services.
My name's Rachel, and in 8 years, I'll be an alcoholic.
Kids who drink before age 15 are 5 times more likely to have alcohol problems when they're adults.
I'll start drinking in middle school, and I'll do some things I don't really want to do. So by the time my parents talk to me about it, alcohol won't be my only problem.
So start talking before they start drinking. To learn more, go to stopalcoholabuse.gov. Brought to you by the U.S. Department of Health and Human Services, the Ad Council, and this station.
Lefty-loosey, righty-tighty. Okay, here's the thing about yellow snow. Now I know my ABCs. Next time, won't you sing with me?
Learn how to drive, you moron!
Remember, you're always teaching. Teach carefully. React with rage, and kids will learn it's okay to do the same. Keep your cool, and kids will do likewise. For advice, visit actagainstviolence.org. Brought to you by MetLife Foundation, National Association for the Education of Young Children, and the Ad Council.
We all come together and stand together to serve our veterans. We invest in the latest technology.
We take the time to train the next generation of doctors and nurses. We work together to make sure we heal their bodies and their minds.
This is our mission. More than 300,000 of us working as one, together with families and loved ones. No matter where they live in this country, we'll be there.
We stand strong, united. Stand with us in caring for our veterans.
They are role models and educators. Their work requires a great deal of time and energy for very little pay. Who are these unsung heroes?
Thanks, Coach.
Thanks, Coach.
The simple truth about education-based athletics in California is this. Without a committed team of coaches and administrators, they just wouldn't be possible. School sports, they bring out the best in all of us. This message presented by the California Interscholastic Federation and the California State Athletic Directors Association.
You're listening to 88.9 FM, KUCI Irvine. The opinions expressed on this program do not reflect the views of KUCI or the University of California, Irvine.
You can have lots of fun on the internet, so don't go places you might regret. A gruff has some net safety tips for you. Surfing the net.